All right, so we're going to pick up talking about the process of DNA fingerprinting. You're going to go through a simulation on it in class. We talked about the fact that some things that go along with this. You have restriction, fragment length, polymorphism, PCR, which is polymerase chain reaction, and STR, which is short tandem repeats. So, restriction, fragment length, polymorphism. Basically what we're going to do, and we do this in class and we do it on paper as well as, doing it with actual DNA, we use enzymes called restriction enzymes, and they are used to cut the DNA into smaller fragments. We then separate those and help to use those patterns or those pieces that were created to identify individuals. So, step number one in the process is isolating or separating DNA from the cell. You did that in class. You use grind up basically, or blend as we did, the cells to break them open. You use laundry detergent, or not laundry detergent, but some kind of a soap substance to break open the cell membrane and the nuclear membrane. And then we used meat tenderizer to separate the nucleus out, the DNA out from other proteins. When we suspended or added cold alcohol to the substance, the DNA floated up to the top. The next step in the process would be to take restriction enzymes, and they do have enzymes that they know cut at specific bases. So they add that to the DNA to make shorter base strands. They then sort it by size using electrophoresis. So electrophoresis is a system whereby it is a gel that the DNA is run on, and there's electricity run through the gel. The fragments will separate by size. Those that are larger move slower, so they're towards the top. Those that are smaller will move faster. Once we have that, then we analyze the specific alleles for identification or for a match. So here are some examples of different enzymes. So BAMH1, if you have the sequence GGATCC, cuts between the two Gs. HEY3 cuts between the G and the C. PST1 on there cuts the same way. So I wouldn't expect you to know these, but I will give you where it cuts when we're using this in class or for a test. Second thing that we can do is called polymerase chain reaction. We don't always have a large amount of DNA to begin with. So what happens is, and this kind of deals with going back to DNA replication, they break open the segments of the DNA and they add templates or primers on there that allow them to copy these certain short repeats that we're looking for, so that instead of having a small number, you can have hundreds and hundreds of copies, which makes it a lot easier to run the gel electrophoresis. So PCR is a technique used for making copies of a definite segment of DNA, and it's valuable when the amount of DNA evidence is minimal. Millions of copies can be made from a speck of blood. So once the actual DNA is cut with enzymes, then the PCR is used to keep copying it like a copy machine, so that you have a larger number of DNA fragments to run on the gel electrophoresis. So in PCR, number one, you heat the DNA strands, which causes them to separate. Second thing you do is you cool the mixture and you add a primer. It's a short sequence of bases that will add to its complementary base pair on the DNA. And we talked about the fact that A bonds with T, C bonds with G, so you always are able to build the other side. Next thing is that they add an enzyme called DNA polymerase, and DNA polymerase is the enzyme that's responsible for copying DNA. So once that primer is in place and then you have DNA polymerase added, the DNA starts copying the rest of the DNA molecule. So you put that, you also put an extra nucleotide so it will bond, and the free nucleotides will bond to the separated strands, and you heat it again for completion. Outcome is doubling the number of DNA strands. Heating and cooling and strand rebuilding is repeated about 25 to 30 times, and that yields more than 1 million copies of that original DNA molecule. and the cycle takes less than 2 minutes from start to finish. So some advantages of PCR. Number one, minute amounts of DNA can be used and amplified. So if there's only a small amount of sample at the crime scene. DNA degraded fragments where there's only a few hundred base pairs in length can serve as an effective template. Large numbers of copies of specific DNA sequences can be amplified simultaneously. So a lot of what this comes down to is it allows us to have smaller amounts of DNA and be able to use them in a crime scene, or if you have DNA that's older and wasn't necessarily preserved all that well, it can be used. It's easy to do. You can buy kits. We had some actually at school, but with the actual labs that we had that went with them were not the best as far as giving us results. One thing to keep in mind with this that is also helpful, one of the problems with studying and analyzing DNA sometimes is contaminant DNA. Even the individual who is analyzing it has to be very careful because your DNA is on everything. So contaminant DNA, especially if it's not from a human source, such as fungus, bacteria, will not amplify because we're using human DNA. So it does eliminate other types of organisms' DNA in that process, but it does not eliminate the problem of human contamination. So electrophoresis, and this shows you what electrophoresis looks like. It has a power supply. The wells are up at the top there, and that's where you put your sample. When the battery is running on there, and there's a liquid that goes through there too that has ions that helps the DNA to move through. There is a negative electrode at the top and a positive at the bottom. So the DNA moves from the well towards the positive electrode, which tells you that DNA has to be negatively charged because negative will attract to a positive. So the rate at which the DNA fragments move on that gel is dependent on the size. So the longer the fragment is, the slower it moves. The shorter the fragment is, the faster it moves. So you can look at that gel and figure out which pieces are the longest and which pieces are the shortest. So electrophoresis is a technique that is used to separate those DNA strands. The current moves through the gel, and that's what causes the DNA to move because the DNA is repelled by the negative. It is moving towards the positive. And when it moves through the gel, it allows it to separate based on size. Smaller and lighter particles will go the furthest on the gel. Fragments that are the same size should end up in the same spot on the gel even if they're in different wells, and that's why we're able to compare them because we know basically how far down it'll travel, and we know that how far down it will travel is based on size. A lot of times too they will use what's called a standard that they know how big the pieces are so that they can compare the DNA they've run to it, and it gives them an idea of what size it is. So after developing, the fragments can be visualized for characterization. So here's showing the samples, pipetting the DNA. They load the DNA into the wells, and then they observe the bands of DNA that come through. So we're going to do this we're going to practice doing this not this week, but next week, where you're going to load gels, and then we'll do it with food coloring, and then we'll go through it with an actual DNA lab. So next thing they started looking at was short tandem repeats. And when you watched that Ed puzzle, it talked about the fact that we all have areas on the DNA that don't code that have a sequence of bases that repeats over and over. And those areas that are not non-coding tend to have more of those repeats because more mutations happen in those areas. So rather than looking at really large ones like they started out with, they started looking at smaller sequences of those, and those are called short tandem repeats. So basically 2 to 5 bases, because remember the other one was 10 to 80, 2 to 5 bases that repeat themselves in the DNA molecule. Advantage of this, it gives us more discrimination. It takes less time. When you think about it, you're not analyzing the whole strand of the DNA. There's 13 different loci that have those particular tandem repeats, and so they can use those to make a positive identification instead of using the whole molecule. So it requires less time, a smaller sample size, and because of that, DNA is less susceptible to breaking down or degradation. So there are 13 CODIS, short tandem repeat locations within the different chromosomes. So we know they exist and we know what those repeats are. So when we're doing that, we are going to, you amplify it. So you extract the gene from the sample, you amplify it with PCR, separate it with electrophoresis, and then the distance that it migrates to determines the number of that repeat that there are. So here's an example of short tandem repeats. Each person has two STR types for that particular gene. One is inherited from each parent. By processing with STRs from other genes, you can narrow down the probability of it belonging to only one individual. So they take those genes, and they also take genes from those other 13 loci to try to narrow it down to one. All you really need to know about short tandem repeats is the fact that they are 2 to 4 bases that are repeating. It's what we look at when we do the gel electrophoresis, and there are 13 loci that allow us to do that. So here's an example. This is what we're doing is showing it what it looks like on a gel in class, but this is what it looks like on a computerized version where they take those results and they input them in. And so each of the possible alleles are numbered for each loci. So the spikes in there shows you the different size of strands of DNA that were found in there. We are not going to really worry about probability so much. Databases have determined what the probability is or how often a particular allele appears in the population. So increasing the number of alleles on different loci, the probability becomes small. So if we know what the probability is of 2 people having the same loci on one of, or 2 of the same patterns on one of the loci, the chances that they'd have it on multiple ones would be extremely low, which is why they run multiples of those rather than just one area that they know about. Okay, and not sure that works, so we'll keep going on. So there's 3 possible outcomes to this. Number 1 is a match, so the DNA profile seems to be the same. Number 2 is exclusion, so it shows that the person who we are looking at is different than the DNA sample that is found at the crime scene, or that it's inconclusive. So we can't tell for sure if it is a positive match or not. There are 2 different types of DNA. One is nuclear and one is mitochondrial. So nuclear DNA is found in your nucleus. Mitochondrial DNA is found in your mitochondria. Nuclear DNA comes from your mother and your father, is found in the egg and the sperm. Mitochondrial DNA is only from the mother. So when the sperm enters the egg, the sperm nucleus joins with the egg nucleus, so you get the male's nuclear DNA, but the mitochondria from the male is on the tail, and that stays outside of it. So you do not get any of the father's mitochondrial DNA. So the nuclear DNA has 23 pairs of chromosomes from both parents. Each cell only contains 1 nucleus. Mitochondrial DNA is found in the cytoplasm. It is inherited only from the mother, and each cell contains hundreds to thousands of mitochondrial DNA. So there is more mitochondrial DNA, and it can be found in skeletal remains. So there's more of them than there are nuclear. Problem with it, it is more rigorous, it's more time consuming, it's more costly than nucleic. You don't have to know how it occurs, but realize that it is a more extensive process. So I'm going to stop here because I am running out of time on the video.